Hello and welcome back to Electro Academy. So today we'll be doing gel electrophoresis. So this is again the part of your methods in biology. So before we have discussed like how DNA, RNA is being purified. Similarly for protein also we'll study the purification method. So there are many methods are like starting from the lysate preparation. Okay, and then we have got the chromatographic columns. So these chromatographic columns, it will take time for to study in details, and then we'll study like how exactly the proteins or everything will be visualized. Okay, so we'll go accordingly. So first, now we have discussed about how DNA and RNA is being purified. So now we'll study about the gel electrophoresis. So in which Agarose Gel Electrophoresis, in which we'll be talking about the visualization of DNA and of course RNA also. So this is how your apparatus looks like. I'm sure you must have seen in your lab also your Agarose gel electrophoresis. So this gel electrophoresis is your horizontal. Okay, so this is going to be your horizontal. ऐसे रहेगा. So you're going to load the sample and it will run from your negative to positive. Okay, now why it is negative to positive? Obviously your DNA is negatively charged. Correct. So we make sure that the DNA and the other sample or loading buffer what we add is also having a negative charge. So what happens when you apply the electric voltage, then it will run towards the positive. Okay. So this is going to be your the flow of electrons. So what is the physics behind it? Everything we are going to discuss in this session. Okay, so why electrophoresis? Of course, to separate the DNA fragments from each other and to determine the size of the DNA fragment. So this is important. See, to separate the DNA fragments from each other, all this, the size of the DNA is going to help in understanding when you're going to do the cloning. So this will tell you कि अगर suppose you are running the gel, ठीक है, and you have got the marker. So this is the marker DNA ladder. We say DNA ladder. हम ऐसे बोलते हैं इसको. ठीक है, so this is already predetermined size of each range over here. It is given like in the protocol or in the procedure, the pamphlet which is provided with the kit you will get. Okay, now these are also again ready mix. Okay, so directly you have to take depending on the company requirement, either 1 microliter you will load or 2 microliter you will load, not more than that. That is more than sufficient for to get the visualization of the DNA marker. Okay, so once you get, suppose you have got over here. Now, अगर वो well में ही रह गया, if it is present in the well, then you know that it is your genomic DNA, okay, which has not been digested and not able to come out of the pore size. So it is not moving. So it is in the well itself. Now, if it is moving and यहाँ पे कुछ size है, यहाँ पे कुछ size है, then you have got over here, but here it is not there. So from these kinds of analysis, you can understand whether the restriction is being done or not. ठीक है, तो इससे हमें पता चलता है कि आपका वेक्टर जो है डाइजेस्ट हुआ है या नहीं हुआ है, अगर लाइगेशन हुआ है या नहीं हुआ है, एवरीथिंग विल बी नोन फ्रॉम दिस. ओके, तो इसलिए जेल इलेक्ट्रोफोरेसिस आपको बहुत इंपॉर्टेंट पॉइंट है, इंपॉर्टेंट इंस्ट्रूमेंट है और टेक्निक है इन योर मोल बायो स्टडीज. It also helps in determining the presence or amount of the DNA, the intensity of the intensity, okay, in which the DNA is present will also tell you the concentration of your DNA, and to analyze the restriction digestion products, which is now just I have discussed. Okay, so basically we don't go with the intensity based DNA concentration. I have mentioned in the previous lecture also, like how to understand the quality and the concentration of the DNA. We go for the nanodrop. Okay, so this will give you the correct measurement कि कितना होना चाहिए. Okay, yeah. So let's start with this electrophoresis. So the term electrophoresis describes the migration of a charged particle under the influence of the electric field. Okay, guys, are you able to follow? कहीं पे doubt हो तो please stop me. Okay, now there are many important biological molecules such as amino acids, peptides, proteins, nucleotides, and nucleic acids. They all possess ionizable groups. Therefore, at any given pH in a solution, as electrically charged species, either cations or anions. So because of this positive and negative, under the influence of electric field, these charged particles will move either to the cathode or to the anode. So we know cations are positively charged, anions are negatively charged. Okay, now tell me, cathode positive है या anode positive है?
Ma'am, anode is positive.
Yes, correct. So anode is positive. So your DNA is going to move from cathode to anode. Okay, ये आपका हो गया negative. Cathode is negative charge. Good. So depending on the nature of the net charge, the molecules will move. Okay. Now similarly, in the case of the proteins also. So proteins do carry different charges, but हम क्या करते हैं to make sure that the entire sample of your protein carries negative charge. For this we add SGS. Okay. So SGS क्या है आपका? Again, it's an anionic detergent. Correct? If you remember, it is a anionic detergent. So when we add SGS in the protein sample, what happens? It gives you overall negative charge to the protein. And then in the machine itself, in the case of the protein, it is known as PAGE, right? Polyacrylamide gel electrophoresis. Okay. तो यहाँ पे क्या होगा? Again from negative charge, it is going to move towards the positive charge. But only difference is that in this case, it is going to be vertical. The system is vertical, and when you're going to load the sample, you're loading onto the top of the well. Okay. So we'll discuss this also later. Okay. Now, the equipment required for electrophoresis, it consists of two items. एक हो गया power pack and electrophoresis units. So guys, one more thing, like I'll just mention over here. I don't think anyone is gonna mention that in any textbook also. So power pack and electrophoresis unit, ठीक है? Electrophoresis unit हो गया जहाँ पे you are going to cast the gel. Okay, the terminology what we use is gel casting, जिसमें आप फिर wells बनाओगे. वेल बनाने के लिए जो हम यूज करते हैं उसको हम कोम्ब बोलते हैं, क्योंकि वो एकदम कंघी की तरह दिखता है. दैट्स व्हाई इट इज नोन एज कोम्ब. ओके, या कोम्ब. ठीक है. सो वी आर गोइंग टू पुट इट ओवर हियर. तो ऐसे आप कोम्ब डाल दोगे, उसके बाद आप उसमें अपना अगारोस का सोल्यूशन डालोगे. ठीक है. एंड देन यू विल लेट इट सॉलिडिफाई ओवर देयर. Now power pack unit क्या हो गया? जहाँ पे you are going to control the you are going to control the current and the voltage. ठीक है. So what we do in the case of Agarose electrophoresis is around 80 to 100 volt में हम machine run करते हैं. Now tell me what will happen if I increase the voltage? If I increase the voltage more, what will happen? Yes?
The samples move faster.
Okay, yes, that is one reason. Now what if I increase more? मैं चलो मान लेते हैं 200 वोल्ट पे कर देते हैं. 200 भी नहीं, 300 कर दे रहे हैं. So this is true that sample will the sample will run faster. This is true. What will happen?
The gel will heat up. And
yes, very good. So it is going to get heat up. Now what happens if the gel gets heat up? Obviously it is going to melt then, correct? And then the entire your whatever experiment you have done is failure. So इसलिए किसी भी instrument का एक limit होता है. There is a protocol in every lab. I'll tell you, when I was doing PhD, so basically I was working on PAGE. Okay, मेरा DNA पे नहीं था, मेरा protein पे था. So I used to run this from 100 to 150 volt में. Okay. So later I came to know that I can do till 200 voltage also, so that the electrophoresis will get complete fast. Otherwise it used to take 1 to 1.5 hours. So when I did 200 volt, so in 45 minutes my experiments used to get over. But okay, so this was just the demo. When we increased to 300 volts, so one of my senior, he didn't know. I told him the 200 is the maximum. So once he did 300 volt, and that was like live experience for me also, like how exactly your the gel, okay, the gel, it also got melted, and the entire sample. Now how we understood was that it got melted because that blue color, the tracking dye which we add, so that we understand, right, कि ये हमारा आपका पूरा gel run हो गया है, it is being done, now you can stop the instrument. So that was spread in the entire buffer. The entire buffer became blue in color, which was supposed to be transparent. So then we understood that between the place there was nothing. The gel wasn't there. So we understood that the entire system, it got heated up and the gel melted. Okay, so this was like my own live experience which I shared. Yes, so good. So these things happens, and it is really important to understand the function of power pack and the electrophoresis unit. Okay. Now, electrophoresis units are available either in the vertical and the horizontal. This is for your DNA, and vertical one is for your proteins. Okay. Now, vertical slab gel units of the type shown in figure are commercially available and routinely used to separate proteins in the acrylamide gels. And the gel is formed between the two glass plates, which are clamped together but held apart by the plastic spacers. अभी क्या होता है? पहले कैसा होता था? You need to add these glass plates and you need to clamp it. Okay, abhi aisa nahi hai. What you have to just do is, और सब कुछ अभी automated हो गया है. Now the plates are designed in such a way that they have got the inbuilt spaces over here. ठीक है, इसमें ऐसे already ऐसे space दिया हुआ है, and इसके ऊपर you can put your another plate like this. So what will happen? So there is a gap between these two plates. Okay, now you need to clamp these. So एक already stand आता है, आपको बस उस stand में रखना है, and that's it. Just on the top you can pour your gel, allow it to solidify, and it is ready. So nowadays it's easy. Before it wasn't, because आपको इसको seal करना पड़ता था, कोई सेलोटेप से करता था, कोई agar से करता था. So there are many ways by which they used to seal. It was difficult. Like running one particular page instrument used to take the entire day. But now in a day, within 45 minutes, you can complete the experiment. Okay. Okay, this is how something it will look like. So you can see that this is the tank. ये हम लोग कौन सा mention कर रहे हैं? You can see this is horizontal, right? So here we are discussing about the DNA का agarose gel electrophoresis. So you can see we have got the cooling plate over here. Buffer रहता है, उसके ऊपर आप gel रखते हो. Now in the figure it is not that clear, but your gel should be immersed in the buffer, otherwise there is no point running the instrument, right? It has to be immersed in the buffer. तो आपका buffer भी थोड़ा सा top पे होना चाहिए. Then only जो भी यहाँ पे आपका जो electrical voltage आप जो apply कर रहे हो, it is going to make sure that those ions are able to move from negative to positive. Okay, yeah. And you are going to, yes, plastic comb is placed in order to you can form the wells. So मैं आपको एक video दिखाऊंगी, and in that it will be more clear. Okay. A gel is cast on a glass or plastic sheet and placed on a cooling plate. However, the agarose gels for DNA electrophoresis are run in the submerged in the buffer. See, in the case of your vertical ones also, they are also immersed inside the buffer. The power pack supplies direct current between the electrodes in the electrophoresis unit. Now, of course, it is carried out in an appropriate buffer, which is essential to maintain a constant state of ionization of the molecules being separated. तो हम इसमें कौन सा कितना pH रखते हैं? We keep it as a near around pH 8. Okay, तो आप जो बफर बनाओगे उसको TE बफर बोलते हैं, या कुछ लोग यहाँ पे TAE बफर भी बोलते हैं. So depending on the lab, you are going to make. कोई आपका ट्रेस EDTA बफर रहता है. Okay. Now, in order to understand fully how charge species separate, it is necessary to look upon the simple equations which is related to electrophoresis. इससे ज्यादा आपको कोई नहीं पूछेगा, ठीक है? तो इतना हमें याद रखना ही होगा. So when a potential difference is applied across the electrodes, it generates a potential gradient. Guys, just let me know, okay, if you are not able to follow. So you have got the two electrodes. So we have got cathode and anode is there. Okay. So when the potential difference or voltage in simple sense is applied, in between these two, a potential gradient E is generated. Okay. Now, since it is having at a distance d between the electrodes, so this is the distance. Now we are going to derive an equation. So when this potential gradient E is applied, the force on the molecule have got the charge Q coulombs, which is equivalent to EQ newtons. So यहाँ पे F क्या हो गया? Force, right? So force is given in newtons. Now it is this force that drives the charged molecule towards an electrode. So यहाँ पे क्या है? We know that it is DNA. ठीक है? तो वो कौन से electrode की तरफ जाएगा? Anode की तरफ जाएगा. Okay. Is this slide clear to both of you?
Yes, ma'am.
Okay.
Yes, ma'am.
So let's move. However, there is also a frictional resistance that retards the movement of this charged molecule. So everywhere this friction is present, हमें पता है, because we don't run the sample in vacuum. Okay, please do understand this, because ये हम लोग interview में पूछते हैं कि why there is always this small fraction of frictional resistance present. अगर ये आपको नहीं चाहिए, then you need to carry out the experiments in the vacuum. Okay. Now, this frictional force is a measure of the hydrodynamic size of the molecule. It depends on the shape of the molecule, pore size of the medium. तो pore size यहाँ पे हम किसकी बात कर रहे हैं? Gel की, ठीक है? Now, in which the electrophoresis is taking place, and of course the viscosity of the buffer. So these are the reasons because of which the frictional force is coming into the existence. क्या क्या है? The hydrodynamic size of the molecule. यहाँ पे कौन सा वो molecule है? DNA. Shape of the molecule का DNA हो गया. Pore size of the medium, that is your agarose gel. Okay, and then the viscosity of the buffer. So these are the 4 reasons because of which the frictional force is coming into the existence. Now let's talk about the velocity by which the charged molecule, here the DNA is moving. So that is given by your V is equal to EQ by F. ठीक है, जहाँ पे F हो गया आपका frictional coefficient. And now we know that EQ क्या है? It is your force in Newton, correct? Okay. So more commonly, the term electrophoretic mobility of an ion is used, which is the ratio of the velocity of the ion to the field strength. So here mu is nothing but your electrophoretic mobility, which is equal to the ratio of velocity by the field strength. Now, when a potential difference is applied, therefore molecules with different overall charges will begin to separate depending on their difference in the electrophoretic mobilities. See, abhi humne bola tha, it is all because of the size and shape of the molecule, right? Now, because the size and shape of the molecule is giving or it is influencing the electrophoretic mobility, that's why the DNA of the larger size and the smaller size, it becomes easy for the instrument to separate them. अभी जैसे larger DNA molecule है तो क्या होगा? The frictional force is going to be very high. Do you agree? See, right now what we saw, velocity is equal to EQ by small f, right? F क्या आपका frictional force है. Now the size of your DNA is large. Correct? So size of the DNA is large because of which क्या होगा? कोई भी चीज कितनी भी बड़ी होगी उसको अगर खींच के ले जाओगे, so there will be of course क्या होगा? It will be difficult to move there because there is so much of friction. आप just imagine करो कि you are taking a just imagine, let us take that you are taking a what to say, there is a anaconda, let us say anaconda ले लिया, ठीक है? तन्वी anaconda को खींच के ले जा रही है और श्रुति एक छोटे से सांप को लेके जा रही है. So for whom it will be easy? Carrying the snake will be more easy, right? Rather than carrying the anaconda. So जितना बड़ा आपका size होगा, उतना ज़्यादा friction होगा. उतना ज़्यादा friction होगा. So what will happen? If your denominator is high, what will happen to your velocity? अगर denominator किसी भी equation में ज़्यादा होता है, what happens to this quantity?
Smaller.
Yes. So this is going to decrease. So that's why longer DNA is going to move slowly. तो वहाँ पे उसकी velocity कम होगी as compared to your smaller fragment of DNA. So that's why at the end we see that larger DNA fragment is present over here and smaller DNA fragment is present over here. Now, one more thing I would like to ask over here. Let us say that ये आपका अगारोस जेल इलेक्ट्रोफोरेटिक यूनिट है, सॉरी ये आपका अगारोस जेल है एंड यहाँ पे हमने डीएनए लोड किया, ठीक है? सो आई सॉ दैट लार्जर डीएनए इज ओवर हियर एंड स्मॉलर डीएनए इज ओवर हियर, ठीक है? सो दिस इज हैपनिंग एट लेट अस से 150 वोल्ट. दिस इज इन 30 मिनट्स रन. Same, I'm keeping everything same, 150 volt, but instead of 30 minutes, by mistake, I kept it for 130 hour. Sorry, not 130 hour, 130. 30 minutes and 1 hour, 30 minutes I want to say. ठीक है? So total 90 minutes के लिए मैंने यहाँ पे रख दिया है. Now tell me, where will you find your longer DNA fragment and the smaller DNA fragment? Question repeat करूं? So I have run the agarose gel, okay, at 150 voltage for 30 minutes. I saw that my larger DNA is over here and smaller DNA fragment is over here. Now, if I'm keeping for 90 minutes instead of 30 minutes at 150 voltage itself, what will happen, or where will I see my larger and smaller fragment?
Ma'am, was the larger fragment loaded there itself? Has it moved?
See, हम लोड तो यहाँ करते हैं ना, यहाँ पे वेल होते हैं, यहाँ पे आपका डीएनए लैडर होता है, यहाँ पे मैंने सैंपल लोड किया है, यहाँ पे मैंने दूसरा सैंपल लोड किया है. अब लोड तो मैं हमेशा करूंगी ना. व्हाट विल हैपन आफ्टर 90 मिनट्स?
Yeah, the larger fragment will still be on the gel, but the smaller fragment
and the smaller
will
fragment
probably have run off of the gel. I mean,
yes,
the smaller fragment would have been off the gel.
Yes, absolutely correct. So what will happen is smaller fragment इतनी देर तक रन कर रहे हैं, so it will run off. So you can find the smaller fragment in the buffer then, which is of course you can't find. Okay, but over a period of time larger fragment आपका यहाँ से हट के यहाँ आ जाएगा. Okay, this is just an assumption that 90 minutes में larger fragment यहाँ आ जाएगा. So what does it says? It says that the movement of the larger and the smaller fragment, okay, it is still happening. आप जितनी देर तक उसको इलेक्ट्रिक फील्ड दोगे वो उतना मूव करता रहेगा. इट जस्ट दैट द लार्जर फ्रैगमेंट थोड़ा स्लो मूव करेगा, बट इट इज स्टिल मूविंग. सो इफ यू कीप ऑन कीपिंग फॉर like suppose I say 90 की जगह मैंने इसको 120 minutes के लिए रखा, so what will happen in that time? Even the larger fragment will run off from the gel. So it is not the gel which is stopping the movement, it is the size of the DNA which is making them to move slower. तो हम क्या करते हैं, इसलिए वी हैव गॉट दिस प्रोटोकॉल डिजाइन कि हाँ बाबा हमें से 30 मिनट्स या 1 घंटा ही रन करना है, अदरवाइज यू वोंट बी एबल टू सी योर फ्रैगमेंट्स. ठीक है तन्वी, इज इट क्लियर टू यू?
Yes, ma'am.
Okay, cool. So these are important. This understanding, this concept is important because आगे जाके जो questions आएंगे ना, it is based on all these fragments only. कोई आपसे ये नहीं पूछेगा कि अगारोस क्या चीज से बना है. ठीक है, nobody is gonna ask unless until you are giving 11th का exam. तो वो वहीं पर ही पूछा जाएगा, otherwise nobody is going to ask. जो भी कुछ पूछेगा यही पूछेगा. And based on this only, if you know that forensic science details also, like how to catch the criminal using the DNA fingerprinting, yeah, how to check for the paternity test, right? So all these things are coming as an application of your Agarose Gel Electrophoresis. Okay. Okay, cool. So, okay, so we have completed this, right? So when a potential difference is applied, molecules with different overall charges will begin to separate owing to their different electrophoretic mobilities. Okay, so even molecules with similar charges will begin to separate if they have different molecular sizes. So they will experience different frictional forces. So in the case of Agarose gel, क्या हो रहा है? Every molecule is having the same charge, right? Everyone is negatively charged. Each DNA molecule is negative charge. Only the difference in the sizes is making them to experience different frictional force. Okay, so guys, is this slide clear to everyone?
Yes, ma'am.
Yeah. So the current in the solution between the electrode is conducted mainly by the buffer ions and the small proportion conducted by the sample ions. So यहाँ पे भी जब आप करंट और वोल्टेज की बात करते हो, so यहाँ पे ओम्स लॉ हो जाएगा, which is nothing but V is equal to IR or V by I is equal to R. यहाँ पे voltage by current is giving you resistance. Okay. Now, so it therefore appears that it is possible to accelerate the electrophoretic separation by increasing the applied voltage, which would result in a corresponding increase in the current flowing. See why? Because it is proportional. अभी हमने बोला Ohm's law क्या बोलता है? That V is proportional to I. So if you increase the voltage, obviously the current is going to increase. Okay. So the distance migrated by the ions will be proportional to both current and the time. However, this would ignore one of the major problems for most forms of electrophoresis, and that is generation of heat. ठीक है, तो इसलिए हम ऐसा नहीं कर सकते हैं कि हम ज्यादा वोल्टेज रख दें ताकि ज्यादा करंट मिले जिसकी वजह से जल्दी सेपरेशन हो, बिकॉज इवेंचुअली इट इज गोइंग टू रिजल्ट इन द जनरेशन ऑफ हीट. हीट जनरेट होगा तो क्या होगा? योर जेल विल मेल्ट. ओके, तो इसलिए हमारे पास कुछ डिफाइंड प्रोटोकॉल्स हैं जिससे which you need to follow. So you shouldn't go beyond 150 or a 200 volt. So during electrophoresis, the power generated in the supporting medium is given by W is equal to IR सॉरी I square R, so current square resistance. Okay, ये याद नहीं किया तो चलेगा, but it is important to understand. Basic physics है, ज्यादा कुछ नहीं है. So most of this power generated is dissipated as heat. So what will happen if there is a heat generation? An increased rate of diffusion of sample and the buffer ions leading to broadening of the separated sample. इसका क्या मतलब हो रहा है? What do you mean by diffusion of the sample? I would have taken the image actually. So what happens is that in the gel, अगर heat हो रहा है, तो आपने यहाँ पे wells में sample डाला हुआ है, ठीक है? So what will happen to this sample? So usually if everything is going at very good condition, okay, ideal condition में जा रहा है, there is no heat generation. So when it is moving, you can see that single band आपको दिखाई देगा, ऐसा move होता हुआ दिखाई देगा, ठीक है? But if heat is generated, then there is a diffusion of the sample. So what will happen? The sample जो होगा वो आपको ना मोटा मोटा blotch दिखाई देगा. So there will be overlap of the sample. So because of which you won't be able to understand कि कितना difference है दोनों sample में, because ये ऐसे spread दिखेगा. You won't have a distinct band over here. ऐसे spread होगा, diffuse होगा. So you won't be able to understand like कौन सा band कहाँ तक है, because see अगर ये आपका 300 है, ये आपका 200 है, ये 150 है, ये 100 है, because of this diffusion you won't be able to understand कि band actual में 300 वाला है या 200 वाला है, ठीक है? So that's why. So this is one reason. Then formation of convection currents, which leads to mixing of the separated samples. So this eventually will result in the mixing of the samples. Okay. Thermal instability of the samples, and rather they are sensitive to heat. Usually they are not, because DNA is stable, and in the case of proteins we always go for the denatured protein sample only. So इतना कुछ effect नहीं होगा unless until you are working on native gel, native page, okay, in which you are going to use the functional proteins or the structural functional proteins. Okay. Then last, a decrease of buffer viscosity and hence reduction in the resistance of the medium. So ये भी आपको मालूम है, कोई भी चीज को जब ज्यादा हीट अप करते तो उसका जो विस्कोसिटी कम हो जाता है, विस्कोसिटी कम होगा, फ्रिक्शन कम होगा, एंड बिकॉज ऑफ व्हिच व्हाट विल हैपन, रेजिस्टेंस कम होगा एंड सैंपल सेपरेशन आपका अच्छा नहीं होगा. Okay, so these are the reasons which can get affected if there is a heat generation. Okay, clear guys?
Yes, ma'am.
So if a constant voltage is applied, the current increases during electrophoresis owing to the decrease in resistance, and the rise in current increases the heat output still further. So for this reason, we don't want to have the fluctuations in heating, इसलिए हम एक constant power supply use करते हैं, ठीक है? So that was all about the heat generation का problem and everything. So what is the answer then? So हम क्या करेंगे, एक कांस्टेंट में जाएंगे. अगर फॉर एग्जांपल इफ एनीवन इज सेइंग कि आप उसको स्लो में चलाओ, इलेक्ट्रोफोरेसिस को स्लो में चलाओ, लाइक फॉर एग्जांपल 40 वोल्टेज या 50 वोल्टेज में चलाओ, सो व्हाट विल हैपन, just because it is so slow, again there will be increase in the diffusion which will result in the long separation time. So इसलिए एक proper तरीके से आपने एक fixed voltage आपको use करना है, ठीक है? So that is what is written over here. Same thing. Yeah, same thing is written over here. Guys, you need to join me back now. Okay, so now there is another one more important factor, okay, which is affecting the electrophoretic separation, and that is something known as electroosmosis, which is also electro-osmotic flow. Now this is happening because of the presence of charge groups on the surface of the support medium. इसका क्या मतलब हुआ? Usually it doesn't affect, because right now the most of the companies, they are providing the ऐसे inert material. तो इसका क्या मतलब है? We are talking about the support medium, जो casting होता है जिसमें आप gel बनाते हो, ठीक है? Even sometimes they carry the charges onto which you're going to make the gel. So even they carry the charges. So sometimes even it can also affect. For example, the paper has some carboxyl groups present. Agarose contains sulfate groups, and the surface of the glass walls used in capillary electrophoresis contains silanol. So these can also affect. अब ये देखो, सारे cases में carboxyl हो गया, sulfate हो गया, silanol हो गया, so they all carry the negative charge. So sometimes depending on the purity grade, it is going to affect the movement of the ions. ठीक है. So इतना effect नहीं करता because it is next to negligible, but still. So as in the case of capillary walls, if you see, you can see that there is a negative charge present. Correct? So these negative charge can affect the flow of your positively charged ions, which is present in the sample. क्या होगा? Instead of moving towards the cathode, what will happen? Because of the negative charge, these positive ions will get attracted to these negative charge, right? So जिसकी वजह से क्या होगा? The flow of these positive ions will get reduced, even though there is a electric field applied. Are you getting the point, guys, over here? So these support negative charge or the positive charge can affect the flow of your ions. Clear? Concept clear है? So this is known as electroendo-osmosis. Okay? ये एग्जाम में आया नहीं है अभी तक. Nowadays, anyways, we have got the inert material, so इतना effect नहीं करता है. Okay, so that's all. Not much. Now talking about the support media. So let's talk about the agarose gel. So agarose is your linear polysaccharide. Okay, it is made up of repeating unit of agarobiose, which comprises alternating units of galactose and 3,6 anhydrogalactose. So ये थोड़ा सा important है. This you need to remember. Okay. Now they are made from the seaweeds. Any idea कौन सा seaweed होगा? Or do you think in India the seaweed cultivation is there? अगारोस कहाँ से आता है इंडिया में? Yes. Yes. Anyone? Have you seen agarose gel? It is like white color gel बनता है in the case of age. Have you seen?
Yes, ma'am.
Okay, so you guys have seen. Okay. So what is the name? कभी उसमें देखा है किससे अगारोस बनता है? Or any idea the agar which you use for your microbiology to make the what to say, the slant if you want to make, you know, or if you want to make the plates for your plating or spreading, कैसे करते हो? agar किससे बनता है? The only difference in agarose is the purity, the structure. Okay, no problem. So this gelatin or the agar हम agarose की जो बात करते हैं, it is from all your red algae. ठीक है? So for agar it is red algae, और उसमें भी याद रख लो कौन सा वाला red algae. We don't use all red algae. It is your geladium. Easy to remember because jelly like रहता है ना. So it is geladium and sorry, it is gracilia. Gracilaria. Okay? Clear? Now, even your agaros are.
Even in India, it is made with these.
So these, that's what I'm asking.
They are Japanese, that's why.
कौन Japanese है? हाँ?
Geladium is known as onigiri from Japan. That is why I wasn't sure. Geladium is onigiri, it's from Japan.
Okay.
I wasn't sure whether it is the same thing.
So the thing is that I'll tell you, in India, अगर आपको ये मालूम हो कि you know whenever you go somewhere, you do get that jelly kind of toffee to eat. ऐसे 1 रुपए 2 रुपए का anywhere. You must have seen. ऐसे not the branded one, okay? Local I'm talking about. Wherever you go, you might get this local brand वाला jelly like things to eat. Okay? So उससे आपको idea मिल जाना चाहिए that in India we do have agar. Okay? The problem is with the agarose because it needs high technology to make it more pure. Agarose is the most क्या कह सकते हैं, like it is the most purest form of your this thing, ठीक है? And this is only possible. So what happens is that mainly from Japan and China, okay, this red algae उसको सुखा के उसको dry करके powder करके they you know it goes to US. And in the US, they process, they make it agar, they make it agarose, and then they sell it to the all over the world. Like even in India, If you want agarose, you have to take it from there. ठीक है? So we don't have this agarose ka making unit. We do have for agar, because agar is your basic raw form. Okay, it doesn't need that high purity wala. And if you see, the agar has also got the color in that. ऐसे yellowish brownish color का रहेगा, right? Whereas agarose is the purest one, the white color wala, right? अगर आपने ध्यान दिया है तो. So in India, so I have worked on this seaweed wala also. So like for my PhD work, there was this community kind of work you need to do. ये थोड़ा सा CSAR का एक नया rule था when I joined. From my batch only it started. It is known as CSIR 800, in which you need to bring your scientific innovative thinking for the benefit of the society. ठीक है? तो आपको ऐसा कुछ करना चाहिए जिससे जो लोकल पीपल हैं, जो लाइक बिलो पॉवर्टी लाइन जो आते हैं, उनको हेल्प हो. हाँ, so that is one kind of work we were supposed to do. So I went to this place known as Mars, Marine Algae Research Station. This is part of CSIR only. Okay, so in Gujarat there is one CSIR unit, CSMCRI, in Bhavnagar. I don't know whether you guys have heard or not, but this place, iska branch Mars hai, which is there in Chennai. Chennai भी नहीं, it is a kind of, you know, ऐसे गांव जैसा area है, Mandapam. Okay, so you must have known about that. ये सुना है क्या? The longest bridge of India, Pamban Bridge. नहीं सुना होगा. This is the only bridge which can open so that the ship can go. अच्छा, ये भी नहीं. एक मैं ये बता, राम सेतु सुना है? राम सेतु तो मालूम होगा, the connecting link between the Sri Lanka and India का last. So that is the area, okay?
Yes,
so if you see, just imagine that this is the last portion of India, and यहाँ से ऐसे connecting dot है to Sri Lanka over here, ठीक है? So obviously these regions are mainly occupied by the Tamilians from Sri Lanka also and from the Indian, and it is difficult to differentiate कौन सा Sri Lankan Tamilian and कौन सा Indian Tamilian. So let it be that case. तो इस तरफ से हो गया आपका, एक तरफ आपका बे ऑफ बंगाल हो गया, और दूसरी तरफ क्या है? Oh God, I forgot India map itself. What is it?
Arabic Sea.
Yes, Arabic Sea, correct. So sorry, पढ़ाई के साथ basic चला जा रहा है. हाँ, so you can see over here what I mean I have observed is यहाँ पे कोई ज्यादा distance नहीं है. It was like only two-way lane, okay? Two-way lane था यहाँ पे, and here like people are gathering like tourist area so that they can see the Ram Setu starting and all, okay? So what I've seen over here was that this area, like in 4 months of the year, this side of area was good for cultivating the seeds. तो यहाँ कुछ नहीं होगा, ठीक है? The another 4 months it was very good over this side, but over here it was bad. Why? Just because of the temperature and the moisture content. Of course, temperature ज़्यादा होगा, so it is going to affect the moisture also. ज़्यादा difference नहीं था. It's just the water quality of the Bay of Bengal and the Arabic Sea. So what I observed was, of course, it was really very beautiful. You can actually, you know, from the top itself, you can see what kind of seaweeds are available over there. So there we have seen that Kappa ficus करके एक होगा. You must be knowing, Shruti, Kappa ficus. Kappa ficus. Okay. So this was brought by one of the scientists who was there at Mars. He went to Japan and he got the sample from there. So these are like ऐसे ऐसे French fries जैसे तो दिखते हैं ये. तो एक छोटा सा पीस लेकर आए थे सर वहां से, and he just what he did was he took that as a basket. He tied that basket and put that seaweed into that. And after a few months, what he observed was that seaweed started growing. ठीक है? And that's how seaweed cultivation started in India. तो जो basic agar जो आता है वो उस टाइम से आता है. Like he went to Japan and got this small portion of the Kappa ficus from there. Just Google में search करना Kappa ficus का. So in India they take this Kappa ficus and now they grow it and इसको जो भी सुखा के इसका पाउडर बना के जो अगार जेली किसी टूथपेस्ट में रहेगा, आइसक्रीम में रहते हैं, जो भी आपकी जेली वाली टॉफी होती हैं, or this gelatin which we make it, you know, ऐसे क्या खाते हैं ना हम लोग, jelly type जो रहता है वो जो हम खाते हैं वो सब, it's all because of this. So it was a small initiative which he, you know, he just started simply by taking that small piece of seaweed from Japan and he brought it over here and started doing. Up, see, what was the benefit over here? The benefit was that this Kappa ficus cultivation or the seaweed cultivation gave so much earning to the localites over there. So they started growing this Kappa ficus over there. They are trying to grow the other one also so that the Agaros can be, you know, produced in India, but growing that particular seaweed is difficult in our Indian continent. Okay, it is not possible. So they tried in Gujarat also, they tried in Visakhapatnam, wherever the seashore is there and it is possible for the localites to work over there. They have tried, but somehow it is not working. So till now, only in that area, Mandapam, which I am talking about, it is possible till now. So इसलिए हमारे पास इंडिया में you can get all varieties of agar, but agaros you need to get it from outside only. So we outsource from there. Okay. So this is a small story. अब इसमें हमने क्या किया था? So we just gave them the technology, like how you can do it. Cultivation तो totally depends on the nature. कि temperature, moisture content अच्छा होना चाहिए, and to cultivate you need sea water. So sea water is free, right? What you can do is just filter the sea water, bring it into the lab, and in the lab you can provide that required temperature and moisture. So this is how you can increase the growth of the seaweed. और आपको अलग से कुछ मीडिया नहीं बनाना, you just have to take the sea water. ठीक है, तो वहां पे लेबर वर्क ज्यादा था rather than more of the scientific, but it was done. So this is how the livelihood have started. And if you see, so gents were involved in growing the cultivation since it is 4 months job only. Okay, so this was like a part time for people over there. Rest of the time they used to go for fishing only. So 4 months ये काम करते थे, and ladies they help in the drying and crushing of these kappa ficus, and उनके बच्चे स्कूल जाते थे. So this is how we help them in managing all this thing. It was a really good work. It was really, you know, it's a good experience also. Anyways, let's come back to agaros. कहाँ से कहाँ चलेगी story. Anyway, so what you need to remember is that it is made up of these galactose and 3,6 anhydrogalactose. Okay, so this is what your agaros is made up of. So it is the components of the agar, which is a mixture of the polysaccharides isolated from the certain weight. So now you remember which seaweed it is. Now, agarose is usually used at concentrations of between 1 and 3%. Well, 3% तो मैंने कभी use नहीं किया, and I have not heard also. But till 2% I have gone. Okay. So for your DNA, just to visualize and to see like how much DNA is present or whether it got separated or not, you use around 0.8% of your agarose solution. Okay. Now, 2% हम कब यूज करते हैं? When you are doing your molecular cloning work, because you need to see the small fragments. So जितना ज्यादा concentration of agarose होगा, high the concentration, the pore size will, what will happen to the pore size?
Smaller pore size.
Yes, very good. So smaller pore size. So as you know, in the case of cloning, you need to visualize smaller fragments, right? So उसके लिए हम क्या करेंगे? We need more smaller pore size. So you are going to increase the concentration, and यहाँ पे pore size decrease हो जाएगा. Since you have got here low concentration over here, so the larger DNA, just you have to check whether the DNA what you have isolated and purified, it is in the purified form or not. You just need to visualize, right? So हम क्या करेंगे? Lower concentration of agarose use करेंगे to see like the large stretch of DNA fragments whether it is present or not. Okay, so this is how it is being done. Now here itself another question arises, like how do you prepare all these solutions? Now, so Tanvi, when we say 0.8%, how are you going to prepare in the lab if I'm asking you to make 100 ml? I think we have discussed also this. Anyways, it's okay even if it is a repetition. So Tanvi, कैसे करोगे lab में? If I'm telling you to prepare 100 ml of 0.8 agarose, yes, Shruti, how are you going to prepare?
We'll take 0.8 grams and 100 ml.
Yes, correct. So simple. You're going to take 0.8 gram and first you put into 80 ml. Okay, try to dissolve it and then make it up to 100 ml. Okay, so in the case of gel and all, it is okay even if you don't make 80 ml. So all these are practical knowledge. Usually अगर आप buffer बना रहे हो तो आप 80 ml में लोगे, mix करोगे and make up the volume to 100 ml. Now in the case of gel, what happens is when you're going to dissolve, of course it will just not get dissolved. You need to heat the sample. आपको अगारोस गर्म करना पड़ेगा, then only it will get dissolved in the water. So what usually in the lab we do is हम 8.8 ग्राम लेते हैं, directly हम 100 ml में डाल देते हैं, because during this heating process, like during this dissolving process, it's a few amounts of water gets evaporated. So that's why we make it directly. Okay, good. So these practical knowledge is going to help further. So now the agarose, so this is poured and allowed to cool to room temperature so that it can form a rigid gel. Now the gelling properties are attributed to both inner and intramolecular hydrogen bonding between and within the long agarose chains. So this is how it is look like. So solution state, you can see that they are not prosperous. Okay, now once you heat and you bring it to the normal temperature, what you can see, initial gel formation is there. Now once it gets solidified, okay, this is how it is going to have the pore structure. So जितना ज़्यादा concentration होगा उतना pore size आपका छोटा होगा. Correct? Now the gel. So that was all about the agarose. Now let's talk about the gel. 1 second now. So the pore size in the gel is controlled by the initial concentration of the agarose. Okay. So the large pore size are fanned from the low concentration. ये हम discuss कर चुके हैं. Low concentration means you are going to have large pore size and vice versa. So high concentration means you are going to have low pore size. Okay. Although they are free from charge, substitution of the alternating sugar residues with the carboxyl methoxyl pyruvate and especially sulfate groups will occurs in varying degrees. So this is again depending on the requirement. तो हमारे केस में कैसा है? In the case of agarose gel electrophoresis, we don't use any kind of substitution. हम कोई भी sugar change नहीं कर रहे हैं, ठीक है? Now, if you do this substitution, it can result in electroendosmosis, which we have discussed, because it is going to interact with the flow of the ions, which is present between the electric and positive electrodes. Now the materials required. So gel casting trays, which are available in a variety of size and composed of UV transparent plastic. ठीक है? So this is one method. एक method ये होता है जहाँ पे आप directly देख सकते हो. There is one video which I'll share with you guys in the group itself, which you can watch. So उसमें कैसा है? The moment you add the sample into that and cover it, okay, it's a very small unit, and what you can see is जैसे जैसे आपका unit run हो रहा है, you can actually visualize directly that your DNA is getting separated. Okay, I'll share that video with you. It is there in my mobile. I'll share. Then you have got the sample combs around which molten agarose is poured so that you can form the wells. And then you have got the buffer like the TAE or TBE. So depending on the requirement, whether it's a tris acetate EDTA or tris borate EDTA. Now, so the migration rate of the DNA fragments in both of these buffers is somewhat different due to the difference in the ionic strength. So usually we use TAE, okay, or TE buffer we say. Loading buffer, it contains dense glycerol. Now, why glycerol is added in the sample buffer? So that whatever your DNA is present, when you're going to load onto the well, what will happen? आप अगर ध्यान दोगे तो ये जो well होता है, let us say that this is your gel and this is your well. तो well क्या होगा? ये सारा submerge है inside the buffer, correct? ये पूरा buffer के अंदर है. Now, if you add anything, what will happen? Because of the diffusion, it will come out and it will get spread onto the inside all the buffer, right? So हम ये चीज नहीं चाहते हैं. So for that, what we'll do, we'll add glycerol. So this will make your sample heavy, okay? And because of which, what will happen? Sample directly आपका well में गिरेगा and it will not come out of that well. Similarly, we also add tracking dyes, that blue color, so that you can understand like मुझे हमें electrophoresis unit कब stop करना है. Okay, so it helps in the visual monitoring of how the electrophoresis has proceeded. सो यू कैन सी आपको वोल्टेज है, इलेक्ट्रोड्स रहेंगे, एक ब्लैक एंड रेड कलर में रहता है, combs लगाए हुए हैं, and then the agarose gel is there. So different figures I have, you can visualize that, and one video is there also in the end. And then ETBR. So why ETBR is used? In order to track the DNA. So during our DNA damage, we have discussed about the ETBR also, that it is an intercallecting dye which gets intercallected, यानी कि बीच में आ जाता है, right? So DNA के बीच में आ जाता है, and it has got the property that under UV light it is going to fluoresce, and that's why it becomes easy for us to visualize the DNA separation कि कहाँ, how far it has, it has reached. So that's why we use ETBR, and be careful when using ETBR because this is also a known mutagen. Okay, so you have loaded the sample over here, then you have applied the electric field. So from negative to positive, it is going to move. This is how it is going to move. You can see ये आपका DNA ladder हो गया, ये आपका sample हो गया. Okay, now when you visualize under the UV light, ऐसा कुछ दिखेगा. So that orange color fluorescence is that your ETBR. Now general procedure. So yeah, I mean, we are just going to read once about the general procedure, but it is very simple. आप अपना बफर लेते हो, उसमें आप अपना अगारोस बनाते हो, हीट करते हो, अपना अगारोस उसको सॉलिडिफाई करोगे जेल कास्टर में, and then you're going to make the well also. At the end, nothing is put into the buffer, load your sample, allow it to run. ठीक है? So the gel is prepared by dissolving the agarose powder in an appropriate buffer. The agarose is disposed in the buffer before it is heating near the boiling point. Avoid boiling. See, अब boiling में क्या होगा? आपका bubble formation होगा. Now, in the case of gel, if you see, for example, this is also important, okay? For example, you have made the gel, and during the heating, the bubbling was there, and this bubbling will get trapped during the solidification. अब अगर सोचो तुम्हारा एक जेल है जिसमें बीच बीच में बबल्स बने हुए हैं और ये सॉलिडिफाई हो गया है, do you think it is going to affect the running of your DNA?
Yes, ma'am.
Okay.
Yes, ma'am.
How it is going to affect?
The pore sizes will be different throughout where the bubbles are there. There'll be no friction, but then where there'll be friction, and that'll affect how the sample.
Yes. See, the thing is that it is going to make a gap. जैसे भी हमने क्या देखा कि suppose let us say that your agarose pore size was something like this, right? If in between a bubble is coming, तो वहां पे तो बना ही नहीं. So your DNA was supposed to go straight like this. Instead of that, क्या होगा? अभी it is forced to go something like this, and उसका पूरा path disturb हो जाएगा, or they can get stuck over here itself. Okay, so this is why the bubbles are not supposed to be there when you're making a gel. तो आप जब भी हीट करते हो, like for example we do it, कुछ लोग वाटर बाथ में करते हैं, they bring the temperature till 60 to 75, okay, but it is a long procedure. और कुछ लोग माइक्रोवेव में ही करते हैं, तो हम लोग माइक्रोवेव में जो करते हैं, so we give it ऐसे 30-45 seconds का heat, and then we shake it, and but not that vigorous कि उसमें bubbles आ जाएं. So just simply to allow the mixing of the agarose. Okay, so once it is mixed properly in the buffer, and then it will be poured onto the casting gel. Okay. Good. Then the moment you pour the gel solution onto that caster, put the comb also, so that before it gets solidified, आपका well भी आपका ready होना चाहिए. Now the concentration already given 0.8%. It gives a good separation resolution of for 510 kb DNA. अब हम ये किस लिए करते हैं? See, increase the concentration, तो pore size कम होगा. Pore size कम होगा, तो हम smaller fragments of the DNA can be used very easily. So it can be visualized easily. Okay. See, this is how the caster look like. तो उसने क्या किया, अपना conical flask में agarose बनाया and यहाँ पे pour कर दिया. Now loading of the sample. So you can see that the blue color tracking dye रहता है, bromophenol blue रहता है. Okay, please इसको भी याद कर लो. It is bromophenol blue. In the case of proteins, we use Kumasi blue. ये protein से किसने है? Okay. So those are also mixed with your DNA sample. So ये कैसे करते हैं हम लोग? What we do is, so there are 10 wells. Let us say that 10 are there. So एक हो गया आपका ladder के लिए, Rest all what we do is we add 2 microliter of DNA plus 2 microliter of your bromophenol blue. ठीक है, तो उसे हम मिक्स कर देंगे एंड टोटल 4 माइक्रोलीटर हम फिर वेल में लोड कर देंगे. कुछ लोग का क्या होता है, दे आर नॉट एबल टू लोड वेरी स्मॉल अमाउंट ऑफ सैंपल, सो दे ऐड अराउंड 6 माइक्रोलीटर ऑफ वाटर, जिससे क्या होगा, दिस विल बिकम 10 माइक्रोलीटर. Okay, so then what happens, like as I was saying, there are few people who would like to make it to 10 microliter and then it becomes easy for them to load. So it's okay, you can add little water, not more, of course. Otherwise, it will get more diluted. Okay, so this is how you're going to load the sample. And yes, again, see, for example, here also, it is very, what to say, like you have to be very cautious. ऐसा नहीं कि directly load हो जाता है. So here you have to make sure that you are able to, load properly. कुछ लोग क्या करते हैं, अगर well ऐसा है तो directly ऐसे load करते हैं. Like here you can see this person is loading ऐसे slightly, but then you know that in the water, when you put something like this, because of the difference in the refractive index of air and water, ये ऐसा दिखेगा टूटा हुआ. Okay, so because of which people are not able to focus properly कि मैं well में properly load कर रहे हैं या नहीं कर रहे हैं. So it is all based on your practice. Either load it vertically so that the deviation because of the refractive index is not coming, or with the practice you can also do. So हम लोग इतना used to हो जाते हैं that literally we don't see whether it is straight or slant. We just pour it because हम इतना practice हो जाता है. We know that this much distance one well is present, so we just simply load it. Anyways, that will come with the practice only. Okay. Then electrophoresis already we have discussed. हमें इसके unit dimensions की जरूरत नहीं है. Just understand that Agarose से आपका gel बना है and we are going to have a set of the constant voltage over here. ठीक है? So this is how your apparatus looks like. Now visualization or the staining of the DNA. So of course we are using here ETBR. Nowadays, like since everyone complained that ETBR is mutagen, we should have something safe. So there is something called cyber safe. SYBR, cyber safe. It is again blue in color. It will give you something like this only, but it is not mutagenic in nature. There are other methods also like cyber green, gel red, methylene blue. So these are many other alternates apart from using your ETBR. Okay. So, but basically many people still use it ETBR only. Cheap bhi hai, thoda sasta hai, and easy to visualize also. Okay. Now, and this is the entire procedure. So you have got the gel caster. You're going to load it. अब यहाँ पे अगर आप अंतर देखोगे, so people have loaded over here with the tape. Tape से seal किया हुआ है, ठीक है? But nowadays you have a gel caster in such a way that you don't have to load it. यहाँ पे बस आपको tight करने के लिए screw दिए जाएंगे, so that इसे plate आएगी and आप tight कर दोगे. You can pour it over there. Then you pour it. You allow it to solidify the entire thing. This entire thing. Remove the tape. Keep it over here. Now there are, it just reminded me of one case where one of my junior, what she did was, she forgot to remove this tape. Okay, and directly load कर दिया उसने sample, sample भी load किया, power pack भी on कर दिया, everything, but couldn't see anything, couldn't see any DNA fragment. Can you tell me why?
The tape.
So the tape was not removed. Since it wasn't removed from here and here, so what had happened? जो electric field जो apply हुआ था, it was not passing from here. जो यहाँ पे buffer था, it was not able to pass, because of which the ions which is present in this area, it was not able to pass. तो क्या होगा, जब electrons ही आपके flow नहीं हुए हैं, तो आपका DNA कैसे separate होता? Correct? Only यहाँ पे in the entire buffer it was able to pass, but no, it is not able to pass from here. And जब यहाँ ही pass नहीं होगा, तो आपको जो DNA जो इसके well में present है, it was not able for them to move, and because of which there was no DNA fragment separation. Clear? So these silly mistakes happens when you start doing the experiments. जब नए होते तो ऐसे ही होते हैं. Anyways, so what are the application? You can see about the DNA molecules in the restriction enzyme digestion. आप PCR products analyze कर सकते हो. Later, when we talk about southern and northern transfer, that is blotting, there also we need this Agarose Gel Electrophoresis in order to understand whether it has worked or not. Okay. then it is going to help in the biomolecule separation. Okay. So this is one video I have got from the Biorad, and Biorad is a very good company where what to say, like many of the apparatus like in my lab we have taken from the Biorad only. Okay, so let's watch this video. Let me share the volume.
This video will take you through the process for loading and running DNA samples on an agarose gel. You will be using a mini subcell to perform agarose gel electrophoresis. The subcell has an electrode wire running across the bottom of each end. This provides for an electric current to pass, which separates the DNA fragments in the samples. First, align the gel so that the wells are closest to the negative or black electrode. DNA is negatively charged and will move from the negative electrode through the gel toward the positive or red electrode. Place the agarose gel into the gel chamber. Add electrophoresis running buffer to the reservoirs at each end of the gel chamber, and keep adding buffer until the wells in the gel are covered by at least 2 millimeters of buffer.
See, you can see now the gel is totally submerged in the buffer.
Place your samples to be loaded in the correct order according to the lanes they are assigned to be run in. Check your lab protocol for this information. To obtain a DNA sample, slowly depress the plunger on your adjustable micropipette to the first or soft stop. Holding the plunger down, insert the tip of the pipette into the microtube with the DNA sample. Place the tip close to the bottom of the sample, then slowly release the plunger button. When loading the samples, keep the pipette tip perpendicular to the row of wells. This will reduce the risk of accidentally puncturing the wells with the tip. Lower the tip of the pipette until it breaks the surface of the buffer and is located just above or just inside the well.
So it is remaining into the well only. It's all because of the glycerol added.
Slowly apply pressure to the plunger button and observe as the sample fills the well. Make certain you stop at the first stop on the pipette, pause, then slowly remove the pipette while keeping your thumb down on the plunger. Once all the samples have been loaded, avoid any bumping or movement of the gel chamber. This might result in the samples spilling into adjacent wells. Place the lid on the gel chamber with the terminals correctly positioned to the matching electrodes on the gel chamber, black to black and red to red.
Cathode, anode.
Connect the electrodes to the power supply, again making certain the electrodes match the terminals on the power supply, red to red and black to black. Switch the power supply on, then set the correct constant voltage for running the samples. See your lab protocol for this information. If a timer is available on your power supply, set the clock for the proper time for your run. Press the start button to begin the flow of current that will separate the DNA fragments. At this point, you should be able to see bubbles coming from the wires at each end of the gel box.
So it means it is running properly.
Notice that the positive or red electrode has fewer bubbles than the negative or black electrode. In a few minutes, you should see the samples begin to migrate from the wells into the gel. Notice as your DNA samples run, the dye front moves from the negative electrode towards the positive electrode.
Now. So once it reaches over here, we'll stop. Okay, so what they have done is, they have put the timer of 55 or 60 minutes ka laga diya tha. So it means what, for 1 hour without getting disturbed, it is going to run for 1 hour at 100 voltage. Hum kya karte hain, like sometimes if you're repeating the procedure, so then you know how much time it is going to take. But if you're doing for the first time, and this is your first time, you know, you're going to calibrate or you are going to standardize the operations, then you are going to check in between कि वहाँ कितना run हुआ, ठीक है? Is that much clear to everyone? Shruti, Tanvi?
Yes, ma'am.
Okay, so depending on like what we have learned today, there are around 2 or 3 questions are there. Let's do that. Okay. Okay, so there are few questions, just like, you know, to give an idea like what kind of questions are coming from gel electrophoresis based. So this is your first question. The gel image below shows 7 alleles, 1 to 7 in order of size, with 1 being the smallest and 7 being the largest. Okay. Now, in the sample of 11 individuals, how many times allele 3 is appearing? Second, how many individuals are homozygous? Third, if this gel would correctly reflect allele frequency in a population, what is the frequency of allele 5? Okay. तो पहले हमें कैसे समझना है? With 1 being the smallest. So which one is the smallest one? This is your DNA ladder. Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 and 11. तो इसमें से सबसे छोटा अलील कहाँ से शुरू होगा? Yes. Yes, Tanvi, which one is going to be the smallest one?
Ma'am, like, I'm not understanding like what you're asking.
Question is, see, इन्होंने क्या बोला है? The gel image shows that there are 7 types of alleles are there, okay, in the order of size. 1 being the smallest and 7 being the largest. So from here, ये तो हमारा DNA ladder हो गया. From here to here, which one is our smallest one and which one is the largest one? I'm asking. Then only we can proceed further in order to answer the question. Shruti?
At the first position, 1650 base pair wala.
So that is your largest wala. ठीक है? That is your 7th allele.
Yeah, that is, yeah, sorry, yeah, the largest one. Smallest one is the 11th, 600. Yeah.
So this is your first one. Tanvi, you got it now? So this is your smallest one and this one is your largest one. Okay. So there are how many alleles are there? 7 alleles are there. So this is your 1, this is your 2, this is 3, this is 4, 5, this is 6 and then this is 7. समझ रहे हैं कैसे किया है इसको? So they all belong to the same size. 7 यहाँ पे है, 6 अगर आप देखोगे तो यहाँ पे ये सब they are coming in the same level. Can you see? This one is coming at the same level. Then 4 अकेला है, then 3 यहाँ पे है, 2, 1 and all. Are you able to follow the figure? Let me just remove this drawings. Yeah.
Yes,
yeah, able to follow. Okay. Now tell me. Now let's do the question. So in this sample of 11 individuals, so we saw, right, from 1 to here, it is 11 individuals. How many times allele 3 is appearing? So they have given that 1 being the smallest one. So ये हमारा 1 हो गया. This is 2. And this is 3.
3.
How many times allele 3 is appearing? Yes, it is
3 times.
Okay. How many individuals are homozygous? I have removed the coloring. Yes. Now tell me, how many individuals are homozygous?
Three.
How? What? Which individuals are homozygous? Tell me the number.
2, 4, and
2, 4, and
2, 3, 4, 7.
7. Correct. So when you say homozygous, it means they are going to have the same alleles. Ab same allele hai, so they are going to be visualized as सिंगल बैंड, वो एक के ऊपर एक ही आएगा, राइट? सो दैट्स व्हाई इट इज कॉल्ड होमोजाइगस. अब जहां जहां पे आपको दो दिख रहे हैं वो हो गया आपके हेटेरोजाइगस. सिंपल. सो अगेन, हाउ मेनी इंडिविजुअल्स आर होमोजाइगस? सो आंसर इज 3. So 2, this is 1, right? 2 single allele is there, 4 also one band is there, and 7 also one band is there. So answer is correct? Correct. Now what about the third? If this gel would correctly reflect allele frequency in a population, what is the frequency of allele 5? Frequency निकालना है. कैसे निकालोगे?
Allele 5 appears 5 times, right?
1, 2, 3, 4, 5. Yeah, 5 times. Yes. फिर?
Yeah, and if the entire population has 11 alleles, then 5 out of 11, right? That will be the 8 something. No, that is frequency. 0.8 something.
So Shruti, we're going correct, but the only problem is that 11 allele types बोले, 11 individuals हैं, right? So does it mean the total number of alleles are 11?
No.
Total number of
One
alleles
second.
over here in 11
It's
individuals.
7. Yeah, sorry. Yeah.
11 individuals. What is the total number of alleles?
7.
How come 7?
It shows 7 alleles, right?
No. One individual means 2 alleles, correct? Let it
Okay,
be
that
hetero.
way. Yeah, okay.
Let it be hetero,
Uh-huh.
let it be homozygous. So 11 individuals means how many total, how many alleles are there?
Okay, yeah. Sorry, because of the first sentence.
So 11 individuals means in the total population. So right now this is my total population, correct? So I'm going to have total alleles say 22, out of which the frequency of allele 5 they're asking. So allele 5 kitne baar hai? This is your 1, this is 2, this is 3, this is 4, this is 5. So 1, 2, 3, 4 and 5. So here 5. Okay. Now it is on you. अगर option में अगर percentage में दिया है, so you can get it like this, or you can give in the decimals. Okay. So how much it is coming? I'll just give you the answer. 1 second. 5 divided by 22 is 0.22. अगर मैं 100 से multiply कर रही हूँ, so I'm getting 22% something. Okay. So this is how you're going to do. So what is the frequency of allele? You need to get out of total number of alleles. अभी हमारे यहाँ पे population क्या था? Of the 11 individuals. So 11 individuals, they were talking about one particular gene. इसलिए they have mentioned only, or they have shown 2 types of allele only, hetero and homo दिखाने के लिए. So even if it is homo, it means what? There are 2 alleles present. Correct? So you need to take it double only. So 11 individuals means 22 alleles. Clear? Unless until they specify saying that this particular gene has got 3 alleles. अगर वो 3 alleles बोलते हैं, so you must have seen 3 bands somewhere in the heterozygous condition, and you are going to have the total 33 alleles. Got the point? Yes?
Yes, ma'am.
Okay, good. Now, this is another figure. So first one is the lane in which, in which the DNA pattern of mother is being given. Okay. And 1 second. Yeah. And in the second and third lane, you can see the DNA pattern of male 1 and male 2 is given. That is, कोई तो एक लड़का है, ये दूसरा लड़का है. On 5th, 4th and 5th, यहाँ पे father का दिया हुआ है. तो आपको ये पता करना है, माँ तो एक है, मगर यहाँ पे बाप अलग अलग है. So you need to tell कौन सा बच्चा कौन से बाप का है. So male 1 का father कौन है और male 2 का father कौन है? Okay. So अभी आप क्या करोगे? Simple hint. You need to compare 4, 5 with 2 and 3, so that you can tell 4th का बाप.
Yeah, 5 and 3.
का बाप कौन है?
4 and 2.
So फोर्थ का कौन सा है?
Two.
Sure. Let's go slowly. Okay. Start comparing from here. So this band is present in 1, 2, 3. Of course, 1 में तो होगा ही, माँ है वो, है ना? So it is present. So जो भी 1 में है, let us ignore for the time being. Now we just need to compare कौन से band either 2 में present है या 3 में present है. So what do we see? ये वाला band present है? नहीं. ये वाला band present है? Yes. Okay. Now what about this band? This band is present here also, here also. This band present here also, here also, here also. तो इसका क्या मतलब हुआ? Guys, are you able to follow? मैं क्या कहना चाहती हूँ? This is present here,
Yeah.
here also, here also, here also. Then here and then here. So what do we see? जो माँ में present है, वो इसमें भी present है. Correct? Then we saw that maximum times the band which is present here is also present here. तो इससे हम क्या समझते हैं? इस बंदे का बाप कौन हुआ फिर? Male 1 और male 2?
Male 1's father is 4.
Yes. So obviously इसका male 2 होगा.
Male 2's father is 3.
So this is how we do. ऐसे एनालिसिस होता है आपका डीएनए फिंगरप्रिंटिंग, जो पेरेंटिंग वाला रिलेटेड होता है. ओके, दिस इज अगेन सिंपल ओनली. जस्ट रीड, इसमें ऑप्शन भी दिया हुआ है. So the parents of a new baby believe they brought the wrong child from the hospital. ये बहुत बार हुआ है, ठीक है. Gel electrophoresis was performed using the DNA samples from the parents and the child. A section of the gel electrophoresis results is shown below. Which conclusion is valid based on the gel electrophoresis result? Mother diya, father diya, child diya. Now you just have to tell whether this child is belonging to these parents or not.
I think it's the right child. I think it's the right child. The bands match at least mother or father, all of them.
ऑप्शन में तो कौन सा आंसर है?
Three.
Okay, let me just give the number. Sorry. A.
Yeah.
D.
C.
So they have the correct child because the genetic information came from the both parents. So if we see, ये भी match हो रहा है, ये भी match हो रहा है. This is also matching. This is also matching. This. Okay, so what do we see? That it is matching with both the parents. Okay. I think yeah, that's all. So this is how you get the question for gel electrophoresis base. Okay, so I hope you got the idea, and this is more than enough to be prepared for your gel electrophoresis. Okay, इससे ज़्यादा और कोई question नहीं आएगा. एक question ये जरूर आ सकता है depending on the concentration of agarose. If you increase the concentration, what will happen to the pore size? What will happen to the DNA fragments visualization? वो एक question आ सकता है for your multiple choice question. Okay, so yeah, good. I'll be stopping here now.